Okay, back to the aseptic transfers. Again, you're just practicing transferring between media. So you're going to get the broth, the slant, and the plate of E. coli, and you're going to be using 3 sterile broths, 2 sterile slants. Basically, you're going to take your E. coli from your inoculated culture broth and transfer from the broth to a broth, from the broth to a slant. And then you're going to take your inoculated slant and you're going to go from the inoculated slant and transfer into a sterile slant. And you're going to then go from the inoculated slant to a sterile broth. And the last maneuver is to go from your inoculated plate into a sterile broth. So by the time you're done, you're going to have 2 new transfers of slants and 3 new transfers of broth, 5 tubes total for each group that'll go into the incubator. Okay. Okay. So again, you're just practicing your aseptic transfers and working with the different media types. So this whole lab is really just some more practice. It's kind of setting you up for most of the media you'll work with for the rest of the semester. So this is what I think of as a lighter weight lab. Just practice. So I like to sterilize my whole loop before I get started. I guess I kind of go back and forth. I really kind of sterilize it twice. I can actually see some pieces of agar on that loop, so I'm gonna really cook it for a second. Okay. Just around so you can see what I'm doing. I'm gonna start with my broth. And again, I'm just going from broth to broth, broth to slant, broth to broth, broth to slant. When you start with the E. coli broth that I give you that's cultured, you're going to want to disperse the E. coli into the broth. It tends to settle down at the bottom. So notice I'm just hitting it in my hand. I'm not inverting the tube because I don't want the E. coli broth to gather up around the top, get all over the place. And I'm not shaking the tube. I'm just dispersing all that gunk that was at the bottom, the E. coli growth, and getting it to suspend back up in the broth column. Still got some down there, so. Okay. We also have a vortex that makes lighter work of what I just did. So you may see me get out the vortex. I don't usually get it out because we only have one. It's hard to put it in a good place, but it lives on our cart on the bottom shelf if you're interested. Okay, I'm just flaming my loop. It's nice and hot. Probably heard it sizzle. I'm gonna cool it to the side. Get nice and cool. Okay. And then I'm gonna get ready to go from, again, my inoculated broth to my sterile broth. And when you pick up the broth, you're going to be able to see it in the loop. Water tension will suspend the transfer in that little circle. And then I'm going to stick it down in my tube and I'm going to kind of shake it around and make sure that inoculate gets into the broth. And then I'm going to flame the top of my vial, cap it. It's ready to go. I'm going to reflame my loop. And then I'm going to do the same thing This time I'm gonna go from my broth to my slant. And again, the general rule is that it's usually easier to go from a broth to a slant, broth to auger. Put this down so you can see what I'm going to do. I've got my broth in my loop, and to transfer, I'm going to go down to the bottom of the tube, and I'm kind of just go back and forth, little zigzags, so that I can streak the broth onto the slant. And you'll be able to see where you've been. You're going to leave little streaks on the auger inside the slant. Make sure the tube is labeled and it is ready to go. Okay. Every once in a while, kind of in between media, I'm going to go ahead and flame the tube that I'm working with, my inoculate, just because that creates a little column of air that prevents contamination, kind of blocks anything from going down in there. And then I'm going to do it when I'm finished working with the broth. Okay, now I'm going to go between my slant and a sterile broth, and a slant and a sterile slant. Slant to slant, slant to broth. So I'm starting with my slant culture. I've flamed my loop. I go down to the bottom. I'm going to get a good smear. You can actually see that gunk, the E. coli gunk on my loop. And I'm going to go into the broth, and just like I did the last time, I'm going to kind of shake it. This is a little easier to see because you get that solid E. coli on the loop, and you'll see the pieces of it jump off the loop and into the broth. And then I'm going to flame the top of the vial, cap it, it's ready to go. Keep them separate. I'm going to flame my loop. 1, 2, 3. And this time I'm going to go from the slant to a slant. So I'm going into my inoculate, getting some E. coli on the loop, and then I'm going to go into my sterile slant and streak back and forth up the length of the whole slant. Flame it, cap it, make sure it's labeled, it's ready to go. I'm going to flame my inoculated culture tube and then put it to the side. Okay, last one is from the plate to the broth. I'm going to flame my loop, get it ready. Okay. Well, actually, I would open your broth first and then open your plate. Have your lab partner open your plate, and then I'm just going to go get some bacteria from the plate, and I'm going to go down into the broth and shake it. You can usually see it jump off the loop. What I have to do is flame the top of the tube, and it's ready to go. Put the lid back on that plate, off to the side. Okay, now I've got all my new transfers. You're pretty much ready to go, except that I would recommend labeling these in a way so that you know which came from which. So these came from my broth, so I'm going to put E. coli on the label, but I'm also going to write broth so that I know that this came from the broth transfer. I'll do the same thing with my slant. Just gonna put like a B for broth, write broth. These two came from my slant. So besides making sure that E. coli is on the label, I'm also going to put an S for slant. S for slant. And then this one came from my plate, so I'll just put the letter P. Your lab manual is going to ask you some questions about which transfers were like easier or more difficult, and so the only way you're going to be able to answer that question is if you know which came from which. And the way that you will measure difficulty is by how much growth you get. So like right now, I just inoculated this broth, so the E. coli population is not very large in here right now, but when I put it in the incubator and pull it out in a day, it's going to get cloudier and look more like this. So I'll hold those up so you can see them together. You can see the difference in the clarity, or the inverse of clarity is turbidity. And so you will be measuring growth by using turbidity. So again, you just want to keep track of which transfers come from which type of media. And then once you're done, you put them in the incubator, and that is the end of the aseptic transfer lab. Until next time, see you.